Okay, so I just turned on the recording, so I'm not like when I was recording my lecture with viruses when I didn't share screen. Okay, so welcome back. We have actually only two more lectures that will be like on exam, well, on the quiz. Whatever is on November 28th will not be on the quiz. And because it's a guest lecture, it will be probably a bonus question on final. I don't really want to torture you to study too much about guest lectures. So usually still didn't decide. Nora will probably insist to give you one multiple choice. And she asked Dr. Kornhauser also. But the lecture that will come on November 28th is not going to be, of course, will not be on the quiz, and on final will be only as a bonus question, which means you don't have to study. But whatever I'm talking about today, and what I'm going to talk about on Thursday, is going to be on the quiz. I don't think that will be hard because most of you already know about all different methodologies like PCRs, and that's what we will talk about on Thursday. So yeah. Anyway, this is my like it's not last lecture. I will have one on Thursday, but I kind of feel I'm already done. So after that week, I will just sit with you over there and be like a student, which kind of makes me feel a little better. So yeah, another thing is that your midterm marks are up. Average is 73%. 72.6, but I kind of rounded up to 73. I was told that that's very high average for third year course, so yay. With two surprisingly is 83 point something. So the way how everyone was, the way how everything went, like let's face it, it was shit show. I thought it will be disaster, but I was slightly relieved. So, and if someone saw extremely, and I had that the day before yesterday, if somebody saw extremely low values for the quiz 2, that can be because you messed up scantrons. Send me an email and I will manually mark everything. I marked manually your 11 questions, and whoever had messed up scantron, like I just ignored scantrons, and I didn't ignore it, but I had scantron and the exam with me, and then I was working on that, so you're not affected with it. But there are people whose scantrons looked perfectly fine, but they got extremely low points, and then they emailed me and I found their quizzes and they got very high marks. So do that, like just if you think you did, let's say so maximum was 50 points, and let's say you got like 20 and you know you studied, just send me an email and I will not immediately, again, really depends where I am, but I will do my best to as soon as possible check your quiz, or find me upstairs on 5th floor of Microwing. Like I'm all the time there. Now I'm most of the time there. That's where I will be because I'm in the lab. Just find me there and we will go through your quiz. But be realistic, please. Like, don't be like, oh, I must be. So you know how much you know. So if you messed up, you messed up. It's fine. But if you know you studied and you checked, you're like, you know whether you were correct or wrong or not, and you see something surprisingly low, just shoot me an email or find me upstairs or text me. You all have my phone number, so you can. Anyway, so we're fine with that. Currently, we have one more quiz that will be easy because there are no Latin names, there are no all sorts of messes. And then we can talk about final later on. Final is not cumulative, yeah, just to know. So final will not include anything before midterm. So, which is the reason why I disagree with transferring the weight of midterm to final. Because I don't think it's fair that somebody just doesn't do midterm and studies 6-7 lectures for final, and then you will have those people who actually did everything but I just don't think it's fair. So whoever wants to do this term, like you have sign up sheets, I am accommodating literally everyone. So that's totally fine. You just sign up there. If room is too small, we will find another spot, but I do want you to do that exam. Okay? Yes, Sid.
Would you be including, or sorry, would you be giving us like a release lab again for the first three?
I won't. And simple reason why I won't is because this is already reviewed. This is way more reduced. So there is nothing to review. So this will be reviewed. So I can just copy paste this in one slide. But I will tell you immediately, the way how I make those questions is I use sometimes Skype. I do. I just copy paste and it gives me questions. So I'm not going to lie. That's how those easy questions are made. If the question is more complicated, that's me. That's literally me. But for those simple, like, so there is no hidden things. Like as you probably saw so far, whatever I tell you, I'm not lying. If I tell you it will be hard, it will be hard. If I tell you it will be easy peasy, it will be easy peasy. And I told you for quiz 2, it will be hard. There is no question about it. It's a hard material. It's hard for me, and God help me for you. And the same how I told you, midterm was not hard. Midterm was like, if you studied, pretty much all those things you already knew. Like we talk about phototrophic organisms in multiple units. So I mentioned that multiple times. I literally told you what will be. I told you there will be a table to fill up. So if you remember, I announced about homolog genes. So you could literally copy paste my announcement, and that was the question. So I'm not going to lie. This last quiz will not be hard. Simple, because the only hard portion will be diversity of eukaryotic microorganisms. I find that one hard. So, but the rest, viruses, methodology, this what we will talk about today, easy peasy. So it won't be. I am more. I have a problem with eukaryotic microorganisms. For me, that's hard. So that's the only thing that I have hard time getting. Okay. And as usual, feel free to talk to me. I do understand more than you think. Anyway, microbes in different habitats. So this is a fun lecture that some of these things we already mentioned multiple times, but this is like a mini recap of everything. And I start to feel like this lecture and the lecture after, like on Thursday, will be something that will be your take-home message from this class. Of course, you will never remember CPV phyla or fat bacteria. Nobody remembers that except me who have to do that. But you won't remember these details. But these are things that you might even know. So we'll talk about habitats in natural and built environments. So microbes that live in natural and built environments. And we will start with natural one first with plant microbe symbiosis. And we already actually talked about many of them. So we talk about microbes that live in association with plants, like with roots. We talk about these root nodules and nitrogen-fixing bacteria like rhizobia. So, but bacteria live in also other parts of plants. These roots, they're the most famous, and that's why I'm emphasizing it. But there are microbiome exists in leaves. It exists in vascular tissue. So it's everywhere. Apparently, it's quite significant for plant performance. So the first, these are three most important interactions between microbes and plants: mutualism, parasitism, and mycorrhiza. And so these are three important types. So we will talk about mutualism root nodules, and we will just briefly mention simply because we already talked about these symbiosis. So this is symbiosis, and this is natural environment. So we know that plant that is participant in these interactions is leguminous plants like soybeans, beans, peas. So those are leguminous. Those plants have natural symbiosis, like it's not something that we forced. It's how they evolve together and they live together. And this they develop this association with bacteria that can fix nitrogen. But those are, don't get confused. That is not cyanobacteria. Cyanobacteria fix nitrogen, but they do not form symbiosis with plants. Simply because they have different ways to protect nitrogenase. The reason why plants develop symbiosis with, or I should say, the reasons why bacteria develop symbiosis with plants is because plants give them protection for their nitrogen fixing enzyme, nitrogenase. But then they have to give something to plant in return, so they fix nitrogen and they feed plant with fixed nitrogen. They live together. That's the point. So, and the fact that those plants can actually grow with nitrogen fixing bacteria actually saves a lot of money for farmers and reduces like the amount of artificial nitrogen that we put in soils, like fertilizers. So those bacteria literally feed plants with nitrogen. Again, just organisms, that bacteria that are participate in symbiosis are called rhizobia. That's common name. But if you remember, there was a genus called Rhizobium. There was Azotobacter. So those are all bacteria that are participating in symbiosis. And plants are legumes. Like this. So basically, rhizobia are fixing nitrogen. They feed plants with nitrogen, but plants provide protection for the enzyme nitrogenase. What kind of protection? Well, protection in terms of oxygen protection, because nitrogenase, the enzyme that bacteria have that fixes nitrogen, is very oxygen sensitive. But plants synthesize something, and I don't have that in slides, and I won't ask, but plants make something called leg hemoglobin. So they just know that they, it's oxygen carrier. I won't ask, it's too complicated. So that's like, they both have a mutual benefit. What this is very this redathezin is very important component that I will ask. So put that in notes. And what is important to know is that actually bacteria makes this compound. It's protein redathezin, and that protein, the name tells you adhesin, adhesin. So helps bacteria to attach to plant roots. So that's what bacteria makes. On the other hand, plant makes organic compounds that will stimulate growth of a very diverse bacterial population in the soil. So, and they both have used with each other. Second type of natural microbial habitat, which belongs to plant mycosymbiosis, is parasitism. And you will find this something that you will see that this is, you will be familiar with this one. So this, who causes gall disease? You had that question on quiz too, Agrobacterium. So that's the famous bacterium that I really like. And there are two of them actually. One of them is causing gall disease, that's Agrobacterium tumefaciens. The other one is Agrobacterium, so same genus, but it's called rhizogenous. It causes a disease called hairy roots. So Agrobacterium is a relative of Rhizobium, but they're not the same organism. And the point is that Agrobacterium is bad for plant. It causes tumors in plant. And the other one, rhizogenous, causes hairy roots. So this tumefaciens, Agrobacterium, carries something called T plasmid or transfer plasmid. So why I'm insisting so much on T plasmid, and I talked about that multiple times. So this is used. So this feature of Agrobacterium is exploited in biotechnology to make genetically modified plants, like resistant to insects and the drought and the various other problems. But initially, this plasmid actually in bacteria, it's in bacteria, and it carries genes that help in tumor formation in plants. And the way how it works is that T plasmid contains gene for opins, and the opins are modified amino acids. Actually integrates T plasmid in plant's genome, and then plant produces opins that feed tumor. So literally plant is doing like a suicide because of this bacteria. But this feature can be very useful to modify plant genetically, but of course you have to get rid of toxic genes. And this is something that I talked about multiple times. So you can modify this plasmid. You have to delete disease genes, and then you keep transfer genes because those transfer genes will help in integration in bacterial chromosome. And then you modify plants to be resistant to insects, for example. So that's and it's really famous, especially herbicide-resistant plants. I think insect-resistant are more popular, but these things are kind of famous, and it's all thanks to the agrobacterium fermentations. Next one is mycorrhiza, and this is the one that I actually know the least about. It is an association between plant and fungi, and because I'm not fungi person and I don't really know too much about fungi, I don't have many things to say, but all what I have to say is basically I just wanted to show you the picture how that looks, how plant root looks without fungal association and with fungal association. But this is actually mutualism. So, and this is important to know. So both plants and fungi help each other. So nutrients go in both directions. It's not like one feeds another. They both feed each other. So it's like fungus transfer phosphorus and nitrogen from soil to plant, and plants transfer carbohydrates. So nutrients just go in both directions. That's how it functions. That's important to know. Again, I'm not really fungal expert, so that's why I don't have many things to say about this. Next one that I wanted to talk about is probably something that many of you might be interested in future, and it is a big deal in microbiology field. And those are microbes in mammalian systems, which are, as you probably heard, microbiomes. So we already know that we have in our gut tons of microbes that help digestion of our food. Because when we eat plant food, plant fibers contain cellulose, and we don't have enzyme cellulose to digest it. But that's why we have microorganisms in our gut that help digestion of complex polysaccharides. And those enzymes are hydrolases and lyases. So what I want you to know is this. There are enzymes that help in degradation of complex polysaccharides. So, and microbiome studies are very popular nowadays with humans, but before humans, what was more popular, and that's something that I really prefer over humans, are herbivore mammals like ruminants. And their microbiome is probably even more rich than ours. Not probably, it is. It's definitely, for me, the most interesting. And because what they have is that special digestive organ called the rumen. So in a rumen, there are all kinds of microorganisms that help digesting cellulose and complex polysaccharides. And what people did, and that this was done years ago, people would just extract 16S rRNA from rumen or caregivers with cows, and then do 16S sequencing. What they wanted to do, they wanted to see what type of microorganisms are there. And they found, you probably remember Firmicutes and Bacteroides. So Bacteroides are very interesting for us. They are not very good for us, right? But they're dominant in rumen, both of them, Firmicutes and Bacteroides. So those are bacteria. Two types of bacteria in rumen, Firmicutes, Bacteroides. But then there are archaea, very ancient one, the first form of metabolism called methanogenesis. So methanogens are archaea present in rumen, which is why cows are actually one of the biggest methane emitters. So they just eat food and they poison our planet. Lovely, yes, fantastic. Anyway, and I have an interesting case study. Like what can happen when you change the microbial community in a rumen? So there's this organism called Streptococcus bovis, and this is the organism that really doesn't like to eat cellulose. So it doesn't like it, right? So if you have a cow that is fed with grass, Streptococcus bovis will not grow there. So grass has cellulose, Streptococcus bovis hates cellulose, no. So what happens often is that you change cow's diet from grass to grain. And why would you do it? Well, grain is, let's say, like sometimes cheaper. And there are all sorts of companies like Dakota Gold Company that makes grain that feeds cows. So you feed cows with grain, so no cellulose. Grain contains starch. Streptococcus loves starch. So then Streptococcus bovis starts growing like a crazy in animals' gut. And because this is lactic acid bacteria, it produces lactate. Lactate reduces pH of the rumen, and that causes acidification, and that leads to the inflammation of the rumen. So Streptococcus bovis will develop in cow's rumen if they eat starch-rich food. If they eat grass, that won't happen. Another interesting case study is when you have when you change the diet. So basically what I'm trying to show you is how the diet changes microbiome. So legume plant, the one that grows with nitrogen fixers, has very high nutritional value, but it contains this mimosine. So that is something that is a chemical compound that has a similar structure like amino acid, but it's toxic. So mimosine in rumen, mimosine can be converted to the toxic compound by existing microorganisms. But ruminants from Hawaii were able to feed with legumes because they had a microorganism that could detoxify mimosine. Australian ruminants did not have those microorganisms. So what they did, they fed Australian ruminants with bacterium from Hawaiian ruminants, and that's how they restored ability of Australian ruminants to eat legume plants. So again, significance of microbiome. The next one is something that is a big, it was a big deal even when I started my PhD. I will be honest, I have never been impressed with that. And that's kind of weird because everyone likes it. Human Microbiome Project. It's really well funded. So apparently, I'm not disputing it's significant, but I'm just not into that. But if you are, that's a great way to start research because funding is exactly there, and just because it has something to do with the human health, as usual. Nobody cares about microbes unless they affect humans, but they do. And there are two major international projects, of course, in Europe and in US. So the center of these two projects is in human health. And it has been established that the microbes that we have in our gut significantly affect our health, and that is why there is a funding over there. So great way to study microbes. So what they were doing when they started these projects, they were just sequencing this 16S rRNA and did metagenomics. And you will see next Thursday how metagenomics and generally omics can be used to reconstruct a basically metabolic map of human gut. There is a highly significant difference between microbiomes in people who are, of course, from different geographic locations. Then also babies that were born like a normally, like vaginal delivery and C-section. So there is a big difference. And also they found the difference between microbiome that are associated with a certain disease. So again, similarities among individuals are more evident at higher taxonomic level, like phyla. Like you will see that, for example, like everyone who eat high fiber diet have more firmicutes, but not in terms of species. So they were not really focused on the type of the species, more on the phyla level, which is great. You don't have to know those ridiculous Latin names. But and this is the example one from the studies. But what happens when you have a low fiber diet? So when you don't eat a lot of fiber, you will have more bacteroides than firmicutes, and the bacteroides will eat internal mucus, and you will develop something called ulcerative colitis. What is happening? So with too many bacteroides, you will see reduction of short chain fatty acids, which is butyrate. Butyrate will drop, and when butyrate drop, that will affect G protein signaling, and will affect histone deacetylase in our organism. And when histone deacetylase is highly active, we get cancer. So apparently, if you don't eat enough fibers, you can get cancer. That's not true, of course, but they associate this high level of bacteroides with a safe level of butyrate and increased histone deacetylation. So, and they also found patterns in various other diseases, and apparently it exists. And this was great study on how you study microbiome. So I will give you we discussed in this sentence. So what you do, you feed people with a certain food. Let's say you eat McDonald's. I want to show how bad food that is, but it's a guilty pleasure. We all love it, right? So you feed people with different food. Then you wait for them to poop. You collect fecal samples, and you extract. You just extract DNA and you do 16S rRNA sequencing to see which type of microorganisms is there, and then you determine changes. Let's say you ate a lot of McDonald's. You want to show that McDonald's is better than Burger King, and you collect poop from people who ate it, but it has to be the same person, and it has to have healthy gut. And this is an example study when what you can do, you can infect cultured cells with intent. So now let's say you know what is internal microbiome, and you infect cultured cells with intestinal microbiome, and you determine how these microbes colonize cells and what kind of inflammatory response those cultured cells are developing. And usually those cultured cells are human cell lines or whatever. I worked only once with human cell line and I didn't like it. And then when you find out what is the response, then you can do transplantation to patients with a fecal sample from healthy persons. So that's, if you heard, called poop pill. So this is how problem with Clostridium difficile was solved. So they implemented to healthy individuals, and they used it to make tablets. And that's how it looks. And you give it to people who have Clostridium problem, and they get better. My husband hates this example, and I adore telling about this whenever we have lunch or something like that. It's amazing to. I was like, yeah, it helps actually. But it's actually very useful, and I'm pretty sure you heard about undergrads told me that each class in this university is talking about poop pill. So I'm not better. Definitely, I wanted to mention because I think it's interesting. I still think I would rather die than take it, but I'm kidding. I mean, I would. Who cares? I mean, we all studied. So. Okay. Anyway, any questions about this? Like we finished with plants, microbe interaction, and mammals, and then we'll move to insects. Okay, so insects and aquatic animals are another type of, so it is some sort of microbiome still, because it lives in insect, those organisms live in insect gut. So that's what I wanted to talk about. So first, insect. So the insect that we will specifically measure are termites. So those are insects that establish symbiotic association with the protists and bacteria that degrade lignocellulose because they eat wood. So they have to have microbes that are able to degrade lignocellulosic matter. If you, let's say that you want to find a new enzyme that can help degrade wood, you will bioprocess there. So the insect gut will give protection to microorganisms, but insects literally get nutrients from indigestible food source. So microbes in insect gut also fix nitrogen and participate in carbon metabolism. And dominant organisms are methanogens, but I would say acetogens are more dominant. So acetogens are occupying termites gut a lot, and they have that hydrogenase enzyme. When we talk about aquatic animals, again, you already saw this when we talked about Vibrio and Alivibrio. So this bobtail squid from Hawaii is small marine invertebrate, and they have this population that luminescent bacteria Alivibrio. So Alivibrio is actually colonizing the squid gut. So, and it lives in that so-called light organ. So those organisms, those bacteria have luciferase enzyme, and they emit light thanks to this luciferase. So that's why this animal will shine in the sea. So, and this is just this picture here is showing you how those alivibrio organisms and vibrio live inside of that light organ. And the molecule, if you remember, that starts all that transcription of luciferase is called AHL. So that's an inducer synthesized by bacteria. It's a quorum sensing molecule. And just again, a little about this luciferase thing. So when squid is young, there will be no bacteria, no alivibrio cells, but alivibrio lives in water, and it will float into the animal using like the animal has those like cilia, and then it will literally grab bacteria from water. And what is surprising is that the only place in animals organism where you will see this bacteria is actually this light organ. And how that works. So the animal produces NO, which is nitric oxide, and that nitric oxide actually kills other bacteria. So that's how it provides a pretty monocultural area in the light organ. So once when light organ is colonized, the level of nitric oxide drops. So this is how they actually establish interaction. Like squid literally helps bacteria to be only culture in their light organs. It's quite amazing what nature can do for you. The last in natural environments are biofilms. And you probably already know so much about biofilms because that is on our teeth. That's probably the most famous biofilms, right? And biofilms are in nature, and this is nothing but organized population of bacterial cells that are attached to the surface, and they're usually found in something like adhesive matrix, and the matrix is made of usually exopolysaccharides, and bacteria itself makes those exopolysaccharides. Shorter name for exopolysaccharides is EPS, and the EPS, like exopolysaccharides, are only one component of matrix. They're very important, but there are also proteins and nucleic acid, everything that builds that binds that community together. And those biofilms are actually made with a purpose to trap nutrients for microbial growth and to help prevent. So they literally prevent the attachment of bacteria from the surface. and why bacteria care so much about biofilm. So it's like a protection, you will see in a sec. So biofilm can be one or two or more species, like it really depends. There are largely diverse biofilms. So on teeth, there will be like 100 and 200 different types of bacteria. So biofilms are very highly tolerant to antibiotics and stressors, and that is the main reason why they are made. So bacteria makes biofilms so they can protect themselves. Once when they are bound together in that community with polysaccharides and proteins and all macromolecules, the antibiotics cannot come in. And that's, you probably heard about Pseudomonas aeruginosa, which you will see later on. It's big problem in cystic fibrosis because it's they just grow in biofilms and then nothing can reach them. So if you can find a way to break biofilm, you can treat with antibiotics, but how will you treat biofilms? And the way how you can treat biofilms is actually if you know their molecular mechanisms. So, and I do want you to know one key molecule about that is related to biofilm production. So when microbes grow in biofilms, they express genes for enzymes that promote attachment. So attachment is opposite than movement. So bacteria can either live attached or freely floating. So the key molecule is secondary messenger called cyclic DGMP or cDGMP. And that is the molecule that is produced by enzyme D-guanylate cyclase or DGC. So, cyclic DGMP actually binds to transcriptional factors, and then those transcriptional factors bind to genes, and those genes synthesize like enzymes that will promote biofilm formation. So in other words, when you have high cDGMP, that means your bacteria will probably live in biofilms and opposite. So if you can find a way to stop D-guanylate cyclase and bacteria cannot grow in biofilm, then you will have easier time to fight against bacteria with regular antibiotics. And those cyclic cDGMP binding proteins are various, and many of them are actually used in assays, which I will tell you in a sec, but I won't add that in exam, just my own experience. So when cDGMP binds to any protein, that protein can bind to the flagellum and then reduces the speed of flagellum motor, so bacteria can't move. So flagellum is moving, and when flagellum is rotating, bacteria swims. But when cDGMP binds to the protein binds to the flagellum is kind of stopped, and then bacteria is living like attached. Or also cDGMP binding proteins can regulate proteins that are required to attach to the surface or mediate biosynthesis of exopolysaccharides. So everything that can promote attached way of life. And again, just an example, there are various assays that are done with those cyclic DGMP binding proteins. So one of assays actually I did multiple times. Basically, I was working with this signaling molecule during my third master, and basically the point was if your cells are producing a lot of cDGMP will bind to the protein will stop bacteria from moving, and then what you see in very like almost liquid agar, you see only one dot, which means bacteria cannot move. But if you don't have cyclic DGMP, then flagellum is very active, and then you see like a big circle of bacteria movement, which means that there is no cyclic DGMP. So those are all like multiple assays are actually developed based on this DGMP binding process. And the example that I have is, of course, Pseudomonas aeruginosa, cystic fibrosis. This is famous biofilm former that is a big problem in hospitals. And this molecule lactone, AHL, that exists in Pseudomonas actually has some gene for D-guanylate cyclase. And then you have D-guanylate cyclase makes cDGMP, then cDGMP promotes biofilm formation, and you have those biofilms in your lungs that actually really affect you. So elevated cDGMP is what promotes biofilm formation and what is a big problem. And why would bacteria form biofilms? They would do it for the self-defense. So they don't want to be killed by antibiotics. So they will tend to organize in the community. They will tend to make a lot of polysaccharides, a lot of proteins or nucleic acid, everything that will make them attach and live together in that closed matrix and prevent antibiotic or any stressor to come in. Another reason is to attach to the favorable niche like basically to stuck where there are enough environments, or to get into the close association with kins, because in that case they can facilitate nutrient exchange between same organs. You saw that in myxobacteria when they formed that using both. Any questions about natural habitats before we move to the built environment? Anyway, so all that I was talking about now was something that is natural, but there are those environments that we, not purposefully, but sometimes very often accidentally build. And some of that is quite harmful, some of that can be useful. But the first one that I will cover very briefly is acid mines. So mineral recovery and acid mine drainage. So in those areas, you will see different types of microbes, and you will now, when we start talking about that, you can already remember this organism that you had on your midterm, peroxidants. So it's an organism that oxidizes iron. So if you remember, microbes can be good and bad for this environment. And what is good is that you can actually, no worries, come on in. I will give an example to you. I don't know. So whoever comes, I am asking. I don't care. Basically, microbes are used to for something called bioleaching. So they're used to extract valid metals like gold, for example, from gold mines. So basically, if you have those low-grade ores, you can extract gold via microbial activities. So how? Well, those areas, ores that have like metals like gold, so they are literally energy reservoirs for microbes. That's one of the ancient metabolism, if you remember from the first lectures, like in the beginning of the semester. So iron sulfide, so iron 2+. It's very abundant. And then you have metal sulfide hydroxide complexes over there. So metal is trapped there. So how will you get it? Well, you treat it with this acidobacillus ferrooxidants. I cannot even pronounce it, but it's ferrooxidants. It oxidizes iron. And basically, that one oxidizes iron and reduces those metal complexes. And then you can extract your gold. So gold is very often associated with something called arsenopyrite. So iron arsenic. So how will you get gold? So you treat with this ferrooxidant organism. That one oxidizes iron. When iron is oxidized, so from 2 to 3 it releases gold. That's what you need. The other one that can be treated with this organism is uranium, because ferrooxidant can actually, in addition to iron, oxidize uranium 4+. And when uranium 4+ is oxidized, it creates 6+. In the same time, oxygen is used as terminal electron acceptor, but this is one of the modes to detoxify uranium. Again, ferrooxidants, quite important organism. The next one, which is this bioremediation, is I almost ended up doing that. I fall in love in bioremediation after I took microbial physiology for the first time, and I had an idea of recovering highly intoxicating areas with benzene, ethyl benzene, toluene, and xylene. And there were all sorts of bacteria that can do that, and I will tell you why I got so impressed. It was, again, microbial physiology class. So basically, what is bioremediation? That basically means microbial cleanup of oil, toxic chemicals, and other environmental pollutants. And what you need to do to use microorganisms for that is to stimulate activity of hydrocarbon degrading organisms. Because what are hydrocarbons? Benzene, ethyl benzene, toluene, xylene, polycyclic aromatic hydrocarbons. So methane is also hydrocarbon. I mean, I did end up doing methane, but there's so many other hydrocarbons that just wait over there to be detoxified. So anyway, so again, well, you will find these problems, well, in areas close to the petroleum industries because they have a lot of hydrocarbons. So bioremediation is the most successful cleaning up oil spills and because there is not a leakage of hydrocarbons. And this is actually, these are so-called oil degrading bacteria. They grow very rapidly when you supply them nutrients and oxygen. They are aerobic. So if hydrocarbon is non-volatile, then that's oxidized very rapidly. Those branch chain and polycyclic carbons are really not preferable, but they can be oxidized by microbes. And the great example of that, and that's how I actually fall in love in microbiology completely, was when my former professor told us the story. Actually, it was a take-home exam in microbial physiology about Gulf of Mexico and the big oil spill that happened in 2010. So he gave us this question and he asked us to here's a strategy, and that was the end. I knew that this was never. So there was an accident. It looked horrible, which you can see on these pictures. The explosion that resulted in a spill of 4 million barrels of oil in ocean. So what was in that oil? So hydrocarbons, low molecular weight, so methane, ethane, propane. So what happened after, I believe, 1 year, everything was consumed, like nothing was there anymore. So how would that happen? The answer lies in hydrocarbon oxidizing microbes. They consumed everything. So, and finding organisms that can do that, and then plus genetically modified options are fantastic. So you can also do bioremediation of pesticides. We use pesticides all the time, so herbicides, insecticides, fungicides. So Burkholderia, potent organism, that one does dechlorination of pesticides. So I'm saying that because there is a question. So Burkholderia is an organism that is doing pesticide-based bioremediation. So what happens is after they do dechlorination, because all pesticides, most of them have a chlorine atom, so they do dechlorination, removing the chlorine atom, and then they break down aromatic ring, and the goal is to bring down compounds into the TCA cycle, so they can actually use it to generate energy. Another organism that I have to acknowledge, even though they are my biggest competitors, I mean, yeah, I don't like them, but yeah, they're good. It's fine. This is bioremediation of plastic. So if these guys come up with the idea, I have nothing else to do. I will just have to teach. So that would be very bad for me. But yeah, so what they thought, so you know that plastic, like traditional plastic, is non-biodegradable. It can stay for 1,000 years. But in Japan, course, they found this organism called Ideonella sacatiensis. And what that organism does, consumed PET polyethylene tetraphthalate-based plastic. So PET is one of the polymers that is used to make traditional plastic. And this organism actually feeds on plastic. What do you think the problem was? It was zero efficient. It wasn't so, but I sort of refused to read their papers, but I think they came up with a good genetic engineering strategy. I'm kidding, of course. It's a great way to remediate plastics. The last one, not the last one, but well, the one that I probably don't have much to say is wastewater treatment. So in the book that I gave you, there is all chapter on wastewater treatment. I just didn't think it's appropriate for this class to talk about the way how is wastewater treated in communities, because this is microbial based class. So wastewater treatment includes many physical and chemical methods, so it's not really microbiology. I sort of tailored it to be to know how microbes are used. But if somebody wants to know more, it is in the book. I want to ask that. So, what is wastewater? So, wastewater is, so it can originate from domestic sewage or industrial sources. It has potentially harmful compounds. So you cannot discharge that water into the streams if you don't apply some sort of treatment. And treatments are physical and chemical methods, but also microbial, which essentially is bioremediation. So when wastewater enters the plant treatment plant, it's treated in facility, and then effluent water is discharged from that facility. So pre-treatment of wastewater involves many mechanical processes, basically to remove large debris, inorganic matters. But the goal is also many times those toxic compounds remain there, like cyanides or acrylamide. So those toxic compounds can be remediated via microbes. So that's microbial treatment, because that's not something that chemical or physical treatment can remove. Maybe chemical can, but it's then you create additional waste. So microbes are very significant in that, like a second step of wastewater treatment. So primary wastewater treatment that has like a physical chemical methods has very high BOD. BOD means biological oxygen demand. So again, don't bother in knowing what that is. That this is a not completely different story. But because of such a high biological oxygen demand, that means like there are many toxic compounds over there. You need secondary treatment, which is a series of fermentative reactions that are carried by prokaryotes, like organisms, usually under anaerobic conditions. So these compounds that are there, like that can be toxic, are fermented, and they'll yield fatty acids, hydrogen, carbon dioxide, and then you can. So this, if you treat this wastewater with microbes, like a secondary method, you can discharge it into the rivers, but this is not potable, which means it's not safe for human consumption. What do you need to do in order to be able to use that water, it's possible to be useful for humans? You need to do that UV, like to get rid of. So you literally need to get rid of pathogens. So you first use microbes to get rid of toxic compounds. Now we have to kill pathogens over there. So, but my point of this saying is that microbes are very significant in wastewater treatment as well. And last but not least is something that we already talked about, and this is just basically a few sentences about corrosions that are formed by microbes. So microbes are highly responsible on corrosions on bridges, which looks very ugly, and it's here. So MIC, or microbial influence corrosion, who is responsible? SRBs, sulfate-reducing bacteria. So organisms that breed SO4 2-. These organisms, as we already learned, extract, they can use organic compounds, or they can use something else. In these cases, they can use hydrogen or iron as a metal. So they oxidize compound like iron, metal, or organic compound, and then they reduce sulfate. When they reduce sulfate, they make sulfide, H2S. Sulfide has highly corrosive nature. And the organism that does that is the sulfopila corrodens. Again, keyword corrodens causes corrosions. So that's what I want you to know. And that will be all from this lecture for today. So as I promised, nothing too hard. Next one will